I don't know if you've spent time with chickens versus pigs. So the pigs are really nice because they're friendly and they get to know you, and the chickens just never give a crap about you. Like they never care. They never get friendly. So they're way less fun to work with.
Everyone, all backgrounds, all ages, all genders, and all walks of life deserve an education.
It is
Come view
my
one of
absolute honor to introduce tonight Dr. Leandra. She has her PhD in animal behavior. She is also a partnered Twitch streamer. I've had the pleasure of meeting her in person, which was actually multiple years now, I think. Thank you so much, Leandra, for coming on the show today and featuring in our Women in STEAM series.
Of course, I'm happy to be here.
So, you're known as this animal behavioral scientist, right? Yeah, that's what your degree is at. What does that mean? What do you study? How does one get involved in that? And where did that all begin?
Okay, let's roll it back. Not behavior, more nutrition focused. So I started, I always knew I wanted to work with animals, right? And I guess as a kid, I wasn't really sure what all of the opportunities available were. I just knew animals. And I think the thing that most kids maybe think about is vet. So off the hop, that's what I thought I wanted to do. I thought, yeah, vet school, that's for me. So I did all like the kind of normal things leading up to that. I volunteered at vet clinics. I worked at a couple wildlife rehab centers, just getting in as much animal work as possible, and then enrolled in a Bachelor of Science program in animal biology, which is pretty general. But also it was at an agriculture focused college. So there was a lot of work geared towards agricultural animals. So I got a lot of that background. And then came the point where I was like, all right, it's time. It's time to apply to vet school. And as a part of applying to vet school, you need some faculty recommendations. So I went to one of these professors that I had that I thought I had pretty good rapport with, really enjoyed her class. And I asked her if she'd write me a letter. She said yes, which was great. Turns out I didn't get into vet school, but she came back at me with, hey, I have this master's program. Would you be interested in doing this? So she pitched a project to me, and kind of that's how I got into grad school. A little bit accidental, a little bit not really the original plan, but that's how that went.
The background story before you had that decision to make is really, really cool to me. Of like, it sounds like you had a lot of different experiences and working with animals that were, I guess volunteering or basically it sounded like anything you can get your hands on to get involved. How did you navigate, I guess, your time and just like where to pick and where to put that effort into? Because it sounds like a lot of work, and also it sounds like you have to be really, really passionate about it too.
Yeah, it's a great question. I was also just the busiest person. Like I was the kid, the teenager who had things every night. Like I was skating, I was bowling, I was playing volleyball. I did all of the things. So to add on top of that kind of academic aspirations certainly was a lot. Luckily, I had really great support from my family to do all these things. But as far as how I picked what I wanted or where I wanted to work or how I wanted to get animal experience, I guess the first place I hit up was just the vet my family used. So I volunteered with them. And for a number of years, I actually got to watch my older dog get spayed, which was kind of neat, but also maybe weird for a teenager to be watching a surgery. And then I guess the wildlife rehab opportunities I searched for online because my initial interest was more in wildlife, and I thought zoo vet might be a thing. So I sought out those opportunities. One was an internship, and one was an actual like summer job. But you just gotta look; they're out there. They're out there if you want to get the experience. You just gotta ask questions and see what's out there.
What was at that stage the most interesting either animal that you worked with or procedure that you saw?
Ooh, ooh, good question. I mean, I saw a lot of North American wildlife. I think one of the coolest cases that we got, which unfortunately was outside the scope of what we did. I was in Washington State, so they're right on the water there, and we had a seal pup come in. And now we didn't have the facilities for that to care for a seal pup, but it's the cutest heckin' thing. And I think there was nothing really wrong with it. It was just orphaned and it needed kind of rehomed. But I'd never touched a seal before, so I got to hang out with him for an afternoon, and that was really fun.
What does one do when a seal pup shows up? Like, I guess you get called, but then what does one do like that?
So I think what we did, we set up a room for it, a lot of like damp towels, and just made it soft and like kind of a wetter space and a warmer space. And it was more just a monitoring job at that point while we waited for um, a marine rescuer to come and pick up this animal. So it was really just like babysitting, make sure nothing bad happens, but also take some cool pictures with a seal.
Nice. So you're almost like the first responder unit in that case, and then the specialist comes in to take care of it.
For sure, someone with the expertise and the facilities to be able to give that animal what it needs.
That must have, like you said, you had to have had a huge breadth of animals that you had seen like those during those experiences. If that's like that intermediary role, right? It's just that's really, really cool. Like I also love animals, so it's so exciting to hear someone who actually went and did it, right? Can be with
Yeah,
the animals and be taking care of them.
I think it's especially in wildlife rescue, it's all about passion, right? Most people there are volunteers. It is largely donation funded. It is very hard to get a person to care about a raccoon because usually they're seen as pest animals. So to acquire funding to care for a raccoon to put it back in out in the wild is maybe not something everyone would be interested in, but really great experience. And if wildlife rehabbers didn't exist and didn't do this work, I mean, no one else is going to help those animals, right?
I In Hungarian, the word for raccoon is a hand washing bear. And so I feel like the name of it is just like it's more fun. Like when I was a kid, it was like, oh, the hand washing bear, that's the coolest little animal ever. But you're right, like the it's something maybe we don't think about for these animal rescues is where does that funding come from and just the image of the animal, right, in terms of the public eye. Like I know there's a lot of thought right about opossums as not being cool animals and yet they're cancer resistant and they eat ticks and I wish that it was also like
Oh
easier
yeah,
to justify rescuing.
they're good little animals. I really like opossums.
So so you're continuing on, you're going into undergraduate to study, for vet school with all these past experiences, and then like you said, you had to pivot and go to a master's program. What was that research project that you were pitched where you're like, all right, this sounds cool?
Yeah, so it was a project that had already been funded, so that was a bonus. But it was with early weaned pigs, so hard pivot into agriculture. Um, Looking at how can we feed these pigs better to decrease the stress of weaning. So the idea is that thinking about animal production, your sow or your mama cow isn't making you money unless she is getting pregnant again, right? To make more piglets. So in on farm in industry, piglets are removed from the mother earlier than they would be in the wild, to be moved on to eat on their own so that they can prepare the sow for rebreeding. And because we do that as early as 3 weeks ish, those piglets aren't quite as developed as they maybe should be, to be transitioned onto solid feed, which is very hard on their guts, right? If you think about it, and you're used to drinking milk and digesting milk, and then a switch into like, oh, what is this soybean meal and grains, and what is all of this? My stomach has never seen that before, my intestine has never processed that before, so it's a difficult transition, and also often leads to a growth lag within these animals. So they are either off feed a little bit or just taking time to build up the proper microflora in the intestine, or to develop the proper enzymes to digest the feed properly. So what my work did was look at a specific supplement to add into the diet of these piglets after weaning to try and stimulate the physical development of that small intestine to make it more prepared to digest feed.
That is so cool, right? And the implications of that work could be like I mean, huge, right? You could be affecting all kinds of farms that are raising, for whatever kind of food, right? How does one
Certainly.
do that work? So for our Women in STEAM series, right, we've had on folks who've been field work for volcanoes. We've had everything from fruit fly researchers, but how do you work with a pig? I imagine you can't bring them into the lab. And then
No.
like, how do you develop the food? How do you test it? How do you control it? Like, just, it seems so alien. Please take us through.
Okay, so as far as the animals, they're all housed at a research farm that was owned by the university. So we were lucky to have access to great facilities that would house these animals. Um, as far as how do you measure it and what do you do? So we had a very specific protein in mind that we wanted to incorporate in the feed. It was called epidermal growth factor, or EGF. And this was chosen because this is a protein that the piglets would usually get in their mother's milk. But because they're getting removed, they're getting less of it. This protein acts in the small intestine. It binds to a receptor there, EGF receptor, where it stimulates the development of the small intestine. So if you think about the walls of intestine, you could think of it like this, right? Like this is the inside, but you really want the fingers there for the increased surface area, if that makes sense, the papillae, where all the digestive goodness happens. And so increased surface area, more room for digestion, more digestion. And the way that we got all of this EGF is actually a whole other part of this of my PhD, or it was a master's. It turned into a PhD eventually, and it was very much microbiology focused. So we had a bacteria, a lactic acid bacteria, which occurs naturally within your gastrointestinal tract, or at least within the one that of the pig. And we genetically modified it to produce this protein for us. So we effectively put a circular plasmid of DNA, put it in this bacteria, and then used it like a factory and did very large scale fermentations of this bacteria, to make, like, I don't know, to grow lots of it so that it would make lots of EGF. And then we isolate the protein from that fermentation. And we took that liquid, tested it, quantified how much protein was in it. And then that is what we mixed in with the solid feed for the piglets. That's how it was delivered.
Very naive question. When you're mixing it in, is it you're just squirting from a pipette?
So think bigger scale. So imagine you have a bucket of feed, which maybe feeds a pen of pigs. Mix that, I don't know, like I don't know a weight, like a bucket, just a regular bucket, and then probably mix in a liter of the liquid, stir it up, and then dump it in the trough.
So it's literally just like the slough of what the bacteria have produced. Like you're separating out the bacteria from the protein. It's just like in bucket loads, like you can really upscale for like the visual part of this and then give it directly to the pigs. And then I presume the next step would be to see how the intestinal development worked. Or was that earlier on, like in cell culture, that you looked at to figure out dosage? Like, how did you figure that component out?
So we did do some cell culture initially, and a lot of Western blotting. So Western blotting is an assay that you perform to quantify proteins. So look at we wanted to know exactly how much protein was being made by this bacteria, and then also be able to quantify how much was within the culture. Um, there was another step of optimization. If you can optimize the codons coding for the protein for the specific bacteria that's making it, that will improve your efficiency. That was another step that we took in the middle there. But yes, before we took it to the animal, we did a lot of bench work with cell culture, adding this EGF to the cell culture, looking at cell proliferation with that, before all before moving to an animal trial. And then of course we did a pilot trial first, kind of a proof of concept, before upscaling to, oh gosh, maybe a 300 pig trial as our first large scale thing.
So, with it being EGF that you're putting into the feed, we've heard of growth factors being out of control in cases of cancer. And it's interesting, right? You're targeting the cells within the gut that are already proliferating fast. Was there any worry about a potential side effect of just overgrowth, or because it's in early development, we're kind of bypassing that possibility?
That was never something that we talked about. I think based off the fact that this is a factor that they would normally get from the sow's milk, and we were feeding it at a concentration that they would normally receive it from the sow had they not been weaned so early. That was not a thing that we talked about as a worry.
Cool. So it's like the biologically relevant dose that you're giving them.
Exactly.
Nice. What,
Yeah.
when you're doing your pilot and you're trying to figure out is this going to work or not, what did that mean for you? Like, what were like, yeah, it's again, it's just such a cool concept to me to work with an animal that has like this direct implication to like world food sources, right? That you're making a difference in. What did your day to day look like with that? You mentioned there was a research like farm that people could go to, or that was run by the university. Did that mean that you did not have to go feed the pigs, or were you like getting the pigs, collecting specimens from them? Like, what did you do, and then what did you analyze? Like, please tell us all of it.
So yes, it was run by the university, but yes, I was also going to the barn multiple times a day. We were giving this supplement both in the morning and in the afternoon feeding. So I would get up in the morning, go to the lab, stop the liquid fermentation of the bacteria, centrifuge it, so spin it down to take the bacteria out, collect the supernatant, put it in a big jug. We also had a control, which was just a blank bacterial media without any growth in it. I would take those big jugs to the barn, and then I would start weighing out feed for each pen, add in the liquid depending on which treatment is in that pen, mix it up, give it to the pigs, and then step away for the day. And do that twice a day. And then throughout the trial, there were multiple time points where we would take samples. And yes, that does mean we would sacrifice some animals. We also had days that were just weigh days through the trial to see how their growth was. We also looked at fecal consistency, so diarrhea. When we did have those sample collection days, we took so many samples. We took almost everything, just because we didn't want to waste the animal's life, right? We wanted to respect that this animal, gave its life for science. So while we certainly had a plan for what we definitely wanted to do, we took extra samples. So we took a lot of intestinal samples. We measured the intestine. We weighed the intestine, both full and empty. We took samples for gene expression. We took samples for histology. We took blood samples to look at some biomarkers within the blood. I think we took spleen, just a variety of organ samples as well, to look at some gene expression and see how we were making a difference with this supplement.
That's so cool. It still just blows my mind that one can work with, because it's not a fly, it's not a yeast, right? It's a pig, and you're talking about these otherwise like experiments that are optimized in these other organisms, and you're figuring out how to run it in a pig and getting these really complex answers. Did you did you ever hear any like I guess complaints from the model organism people about how much harder this might be, or was it where the methodology and you just had it optimized where it's like, I got this. This is not a difficult thing to do.
No, I mean, there was well-established protocols both from the lab I was in and also the research farm, right? And everything that you do with animals has to be justified and approved by a committee. So no one is in there willy-nilly doing experiments. Everything has been run through the correct checks and balances to make sure what you were doing is appropriate, and then also checks on the number of animals that you're using and the number of animals you're sacrificing, and to make sure that you are using enough to get statistical significance but also not overdoing it, right? So yeah, everything is checked.
What what were the ethical considerations when working with these animals? So we've had comparisons, right, where with insects it's just kind of you do whatever you want. With mice, right, there is these IRB approval boards and you have to, like you said, justify everything you do, but also certain kinds of living conditions and density of the animals. What did that look like when it came to working with pigs?
Yeah, so like I said, when you plan out any animal experience at the university I was at, you have to fill out a form detailing almost everything that you're going to do. And that goes to an animal welfare committee where it is reviewed, and either they come back at you with questions and suggestions for what you should be changing, or they approve it. I think the biggest sticking point is perhaps, like I said, how many animals are you sacrificing? And then also your method of euthanasia is really important to them because, and important to me, right? We want to minimize animal stress and suffering. So getting those protocols nailed down is really important.
In your pilot study, did you come across anything surprising that informed your later bigger trial? Or I guess also what did success look like in the pilot? Like where did you say, okay, I have enough data, I'm gonna move forward with this bigger trial. I'm satisfied with X. Like what were the X's that you were satisfied with to go forward?
Yeah. So with the pilot, we actually had three different treatment groups. I know when I spoke about how I get ready for each day, I talked about centrifuging down the liquid to take the bacteria out. We had one group in the pilot where we didn't do that. We just fed them the full fermentation with the bacteria in it as a third group. So control with no bacteria at all, EGF treatment group with the bacteria removed, and then EGF treatment group with the bacteria in it. And we thought that maybe giving this live bacteria to the piglets that was still, potentially actively producing EGF could be additionally beneficial, but it was not. Insofar as growth performance. Largely, we were looking at growth performance as a response, and initially, especially with a pilot, before you go further with samples and do gene expression and I don't know, any digestive enzyme analysis or stuff like that, because that's more money, right? Money and time, and you want to make sure before you invest in that. Um, so what we thought was that by giving them this bacteria, we were actually overloading their systems or overwhelming them. So that was cut from the treatment groups moving forward. And no other animals after that got the full fermentation product with the bacteria in it.
It's yeah, that is a surprising result, right? Where it's like you'd expect if you're putting in the little factories, which the bacteria are for the protein, into the gut, as long as they were functioning, it's almost like you'd have an extended dose.
Yes, that's kind of what we thought, but it didn't work out that way.
So does that mean that when that didn't work out in the pilot for the full study, do you not have that set because you kind of already have an inkling as to that's not going to work?
Exactly. What we did instead, I think after the pilot, was we added an additional dosage value. So we still had more than two groups, but instead of including none of the groups included bacteria, and I think we added a group that got just more EGF to see if that had any more impact.
What did the upscale look like? Like from pilot study to full study, you mentioned the importance of numbers and figuring out how many individuals to test. Do you remember what that looked like in terms of the upscale?
I don't remember exactly how many animals we used, or like, but probably think about 10x likely, which is still not quite hitting. It's not really not hitting a production level, but probably 10x from pilot to full scale.
And how long do you have to run the study too? I guess that was probably figured out in the pilot as well, but like at what point is because you're going to be altering the dynamics of gut development, which presumably is going to make it hard, right, to figure out the end time of like this is how far we're going to take it. Um, how did that affect it? And I guess also just as a side, like what were some of the challenges that you faced in this really really cool set of experiments?
Um, for timing, gosh, how long did we keep those animals for? We probably kept them for 2 months. Um, One thing that is good and bad about working at a university research center is that you share the space. So you both have to wait to get animals in pens, and then also you can only have animals in pens for so long before the next person wants it. So that definitely had some influence. Um, Not to say that we didn't keep it within a biologically relevant time frame, but that was a factor. Um, and as I said, like we did take samples weekly, or not, maybe not samples, but we took weights weekly. So we were following these animals through the whole trial while they were on treatment, and then I believe we had three different kill days. One of them being at the very end, um, just to allow us to follow any developments, or follow the intestinal morphology through them eating this epidermal growth factor. Um, does that answer your question? Sorry.
Yeah, yeah, no, it's a lot of naive questions just because I'm like, I can't even, I've never seen anything like this, and it's just such an alien but cool scientific concept to me, like there being a university farm, right, that you could
Hmm.
do these like research questions at, and it's like it's a whole avenue of research that I think it's really inspiring because like you said, it's kind of it's still a foil to like that vet field that you were going to go into, right? You're still studying the animal welfare and behaviors through this nutritional element to it, and it's so cool and so unique to hear about it. So if the questions are coming across as naive, it's just because I'm just silly excited and just wanting to learn all about it.
No,
Uh,
no, I'm happy to share.
what was I guess the end result of this experimental series, and was there an actionable item that y'all did or tried to or trying to put in place from it?
Right. So I think the idea was eventually for it to be a product, like to be made into a product. And now there was a lot of steps beyond me being there and my part of it before that could be a thing. Um, while I was still there, I did some work around trying to make the bacteria heat resistant, because there's a lot of processing with feed. It goes through extrusion, like an extruder to be pelleted. That creates a lot of friction, a lot of heat. So I did some work to try and make heat resistant versions of this bacteria. But ultimately, um, I think the project that kind of picked up where I left off was transitioning into using yeast instead of bacteria, because we found them to be more efficient factories than the bacteria we were using. So the next project after me switched over to using yeast.
And it's so, but all the processes that you had discovered, that you had learned about, right? Like dosages of feeding and biologically relevant amounts and timelines, like that can all still be transferred independent of switching that organism, right?
Right, still relevant, still very relevant background. It moved into more of an efficiency standpoint of like, because that is kind of everything to do with agriculture, right? Like, it's a nice thing to say that, hey, if we feed this to our pigs, they have an easier time, their health is better, and they grow better. But then you have to turn around and go to a producer and say, it will cost you this amount of money. Are you going to do that? So it's kind of the connection between the cool science and then making it an actual thing that people want to buy into effectively.
It all reminds me a little bit of drug development, right? Where it's like if even if you have this miracle drug, if it needs to be held at negative 120 before the delivery with a needle injector 3 times, right? The likelihood of people buying into it is really low versus if you can just pop a pill and it's stable, it's much more of a
Yep.
higher buy-in. So that's really cool that you're having to account for all these different variables, like with the heat resistance, for like the pelleting. Like you were taking it from the very infancy stage of it to the now the product development side of things. It's did you have a favorite part of that timeline in terms of what you worked on?
For sure. So I know I touched on this briefly about scheduling as far as the research barn at the university. Um, there was one point where we just couldn't get in. Like the barn was booked out for months, and we had this project that we needed to get through. And my PI, the principal investigator, had connections with a feed company in China. So I got to go to China and run a pig trial there, which was pretty darn cool. I was in China for about 3 months, and it was an amazing experience.
That's that's so cool. Could you tell us a little bit about that experience? Like what were some of the I guess differences in terms of the research side of things, but then also what were just some cool cultural differences that you look back and you're like, man, this was really, really fun.
Um, so I'll tell you, I'm wondering if I should tell you like a bad story. I guess one thing that was really interesting and cool was that, so this was an animal nutrition company. They didn't necessarily have any experience in the microbiology side of things. So they effectively built a lab for me there to like test the bacteria and test that it was all working and good to go, which was super cool. And I remember this. So, I mean, in a lab, you wear closed toe shoes, right? That's safe, protect your feet. But they had lab sandals that they had bought specifically to be clean and to be worn in this lab, which was, it blew my mind a little bit. Um, but it was an amazing experience. The language barrier was difficult. I had a translator who was with me most of the time, but there was still some issues with, I guess, some of the farm workers didn't fully understand how a research trial worked. And there was a point in time where they gave some antibiotics to a couple of my animals without asking. This was an antibiotic free trial. They had to be removed. The animals had to be removed from the study. Um, but overall, it was a really great experience. And I always appreciate learning about a different culture. So to be able to communicate with people there about science and kind of show them what I do and learn from them about what they do, it was really great.
It's really cool to have had like that lab built for you, but essentially, right, custom built. Was that intimidating at all that, you're at the time, I'm guessing you were still a graduate student and going
Yeah.
overseas to run a trial and they're like having this lab for you? Like that seems really intimidating.
It was kind of wild because I there was a point where I sent them a list of all the things that I wanted them to purchase. And I was like, this feels like a lot of power for me to have. And then, of course, when I got there, not all reagents are made equal internationally, let's say. So I was trying to test the fermentation product to make sure that, hey, like, yeah, it works. We have EGF here. And for the life of me, I still don't know what the issue was. Like, we could not get their Western blotting to work. Everything was weird. And so we ended up switching to dot blots, which is just another way of visualizing protein, but a little bit less quantitative than a Western blot would be. So it wasn't without its hiccups, but it was fun.
It's every time you say Western, I've got this little cringe in the back of my mind. I'm like, oh,
Oh,
the pains of Western blondes.
terrible, right? And I feel like that was almost my entire PhD was just Western blot after Western blot, the
Well, and then
horrors.
going international, and it's like I've done this so many times. Why is this not working?
Yep.
So, so you mentioned that it transitioned from a master's into a PhD. How did that transition come about, and what led you to choose to continue down that academic route?
Yeah, so it started as a master's. I think like a year and a half into it I transitioned. And it really just happened because I had my hands in so many pots. Like I was working on my project, but I was also like working on a bunch of other little things and helping other people and my supervisor just came to me and was like, "Hey, you've put in more work. Like you are doing more work than you need to do for a master's program. So, like, how do you feel about transitioning this?" And I did really think about it because I still kind of maybe had in the back of my mind, like, oh, like vet school, like after your master's, go to vet school. But then I really enjoyed my time in that lab and I really enjoyed the work I was doing. Um, so it felt right. And I said yes. And we transitioned and it was very smooth. Yeah.
That's really, really cool. And it's really reassuring to hear too, like, hey, you're doing a lot of work more than for this particular tier of degree. Usually, because we've heard of a lot of experience of like, well, you're here, so just work, 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 work. And it's like the seeing that yes, how much work you're putting in, valuing it and saying, you know what, let's make sure we get you rewarded for it with this
Yeah,
upper level
exactly.
degree.
Like the recognition that hey, you should get more. You should like yeah. Yeah, you're putting it in. Let's make sure you get credit for it.
Did you have a favorite side project that was different from the main
Um
one where you're like, this is like a sneaky fun one?
so we did a lot of experimental isolating, let's say. Um so there was at one point a fish carcass showed up at the lab and I think it was a carp. I can't remember. Some sort of fish that like just eats hard to digest things. And my supervisor was like, we're going to open this fish up and we're going to take its intestines out and we're going to isolate the bacteria here because this fish is good at digesting stuff. So we're going to see what bugs are in there and like what is doing the work. Because another angle that we were thinking about for these piglets was like soybean meal is really hard to digest for a piglet. So, is there a bacteria that we can use to almost pre ferment the feed for them? So that's a thing that I did. One day we just pulled apart a fish and took its insides out and tried and isolated bacteria from it.
That's such an incredible day. Like again, it sounds so like in principle, right? It's not that different, right? You're isolating from the gut and you have the tools that you can pull together. But I'm just imagining like, hey, we got this fish today.
Yeah, right. Just sure. Okay.
what was what was your next step? So you've gone and you're defending your degree. You're now Dr. Leandra, right? What did you go on and what was your next step? What were you thinking? What were you interested in? What did you end up going towards?
Yeah, so my next step was actually another thing that was just kind of offered to me. So one of the members of my defense committee worked at a lab nearby, and after I defended, or I guess no, before I defended, he had sent me a message and been like, "Hey, I have this project. Would you like to come work for me after you defend?" So I said yes because I liked him. I liked working with him. It was with broiler chickens, so still a monogastric or a single stomach animal. And it was still working in supplements they were looking at for these birds. So it was adjacent to what I was doing, and so I said yes. So I moved from pigs to chickens.
What was the difficulty in the transition from pig to chicken, or was it one where again, you know all the techniques, it's just they're gonna peck a little bit more than a pig might, and you just go on?
The transition was easy. Yeah, like you said, we did a lot of the same stuff insofar as sample analysis. Uh, One thing that's really nice about working with chickens, especially broilers, with how we have bred them in North America for meat production, is that their full life cycle is about 3 weeks. So you can run through a broiler chicken trial really quickly and get a lot of data really fast. So that compared to a pig. So that was nice.
What was your research question for with the broiler chickens?
With broiler chickens, I was working with a butyrate glyceride product, which is known to influence how they digest fat and also has some antimicrobial action. So I was looking at fat deposition within the muscle tissue and how that changed on the supplement. And then also, none of these birds received an antibiotic. And then, so we looked at how also how this product worked as an antimicrobial as far as animal health.
Was was there what was like the big takeaway, I guess, of that study? And then what were, if any, challenges compared with working with the pigs?
Um, I mean, the takeaways were that it worked. These are products that do exist and are were under development on the time. So I actually did test a product that was being sold. It was more for them to use for their, I guess, marketing, like to say, hey, we've done the science and this is what you see. Um, As far as challenges compared to pigs, I mean, I just don't like chickens as much. I don't know if you've spent time with chickens versus pigs. So the pigs are really nice because they're friendly and they get to know you, and the chickens just never give a crap about you. Like they never care. They never get friendly. So they're way less fun to work with. I know that's very like not sciencey at all, but just on a personal level.
I bet that also made a from that was like a neat part in terms of being able to work with them, but that probably also made it difficult for some of the experimental things that you had to do, right? Was there any, I personally can never work with even a mouse just because I'm worried that like I'd have form that kind of connection with the little
Mm.
furry animal. Um, was that also an element that you had to grapple with? And in the like, compare and contrast, or was it one where you kind of know ahead of time that you just have to be detached, and even if they're a little bit more friendly, it's like I have to view them as this like lab number almost?
Yeah, so it's funny you say lab number. We had a rule with the pigs, um, wherein we weren't allowed to have favorites or name any of them until after the last kill day. Um, so just so that nobody got attached to a single pig, because the selection for sampling animals was done fairly randomly based on weights. Um, So you never knew, you never picked a favorite because your favorite could maybe be the one chosen for sampling. Um, this was not an issue with chickens because none of them were nice.
I My wife has the same take. She kept chickens when she worked on a farm, and yeah, I think there was one hen that was her friend, and the rest were just an absolute no
Yeah.
thank you. So you work with then pigs, chickens, you've done all this development on, I guess, their growth and really helping the animals in terms of like increasing productivity for potential consumption. What was the next step after that? Like, what did then Dr. Leandra decide to do?
After that, I went to cows. So I was just like working my way through the animals. No, due to like some life circumstances, I ended up at a different university, working with dairy cattle, which was a big change because, I mean, they have a completely different digestive system, right? So both chickens and pigs are monogastrics, meaning single stomach, whereas cows are ruminants. They have four stomachs or four different chambers to their stomach that the feed works through, including the rumen, which is a very large fermentation vat full of bacteria that are doing the digestive work for the cattle. So that was a big shift and a big switch in how you think about things nutritionally. Um, and that was also a shift in kind of research theme. I was still looking at nutrition, but instead of growth performance, I was looking at how it linked to reproductive performance. Um, yeah.
That seems like it would be the opposite in terms of timelines from when the chickens, where you could run it in 3 weeks. If now you're looking at reproductive success in a cow, I imagine that's on the order of years, yeah?
Well, so I was specifically looking at time to first estrus. So I was working with heifers who, had never cycled, and how nutrition status affected how quickly they came into estrus for the first time. So it wasn't years for me, not for that kind of thing, but it could be, especially like um, if you're looking through at multiple generations, like through calving and then any carry through effects on calves, like that could be a very long process. But yes, the lifespan of a cow is much longer, much longer than a chicken. Um, Another nice thing about working with cows is that they're very valuable. So I I never killed a cow. Like it was rare for a study to be terminal because they are worth so much. So any sampling you're going to do is usually scheduled or finessed in a way where the animal lives.
Was was the monitoring ever, did you ever get to the point where you got to use this? It's an interesting tool. You got almost a window into the cow's stomach. Did you get to do any of that as you were pulling sample, or was it just more like scraping cells from the gut tissue and more traditional methods?
So my cows personally were not rumen fistulated, which is what you call that. It is a rumen fistula. It's a hole in their side that goes right into their rumen. But another researcher I was working with did have those cows, and gosh, they're so cool. Like it's so neat, especially for like nutrition trials. Like she was just putting in like bags of feed, like mesh bags of feed. She would plop them in and then leave them and then pull them out and see how well they digested. Um, those animals are so neat, but not mine. Mine were mostly set up with jugular catheters. So I was looking at increasing propionate status, which is a fatty acid that they use for energy that largely also impacts their reproduction. So my cows just had needles into their jugular veins, and I was giving them a little extra.
So you were looking at the molecules that were almost preempting when you're going to become reproductively active, like the presumably those fat cells or fat storage levels would increase, and then you'd have follow of like estrogen or other reproductive compounds.
Yeah, we looked at progesterone and how progesterone developed in these animals, which is a known downstream effect of propionate. So we were looking at, hey, it was kind of like skipping the nutrition part almost. We're like, because if you worked backwards from that, the next step back would be, okay, increased propionate status works. Now, how do we design a feed that will allow for her to absorb more propionate? And it's a bit more complicated with a cow because there are so many microbes in the rumen that are producing these volatile fatty acids for the animal who is then absorbing them. So there's like an extra step between just like, oh, I want to feed more propionate. It's like, well, no, I want to feed a forage that breaks down specifically by these microbes into propionate. It's just it's an extra step.
Did you have a favorite model organism in terms of which one you
Um,
worked with? Because it seems like there's pros and cons to all of them. Maybe chickens ranking like some pros there, but emotionally they're maybe on the lowest of the totem pole.
I mean, it has to be pigs. I was with them for the longest. And gosh, they're just like little doggos, aren't they? So the pigs were my favorite. Maybe not. I mean, the chickens are more practical insofar as getting through generations of them for research, but um, they're just not as fun. And cows are good. Cows are nice, especially dairy cattle, which I worked with. Um, they're just like big puppies too. After a while, when they once they figure out that you're the person who brings the food, they're good friends with you as well. But it's got to be pigs for me.
Cool. I love hearing all these like unique organisms that, otherwise I don't think many people hear about this active research that's being done and all the considerations that you have to do in working with each of these animals, right? From the practical perspective of how you do the work to actually doing the work itself, the techniques, the animal welfare. Like you are well versed in a ton of elements of the science and life cycle of these animals. Like it is really, really cool.
Oh, thanks.
So what is Dr. Leandra interested in thinking about doing, working on now?
So I've actually pivoted a bit more into scientific communications. So without being exceptionally specific, I'm more on the journalist side of things, where I am speaking with researchers. I am reading research. I'm looking at what's new and what's coming, and I'm doing more writing. Um,
Still on
yeah.
the on like the dairy agricultural side of things, or now it's just a little bit more general, bit of both?
Still on the agricultural side of things, and still very much related to animal health. Yeah.
Cool. And I have to also pivot a little bit. How does this fit into your Twitch streaming career? Because you're also a partnered streamer on Twitch.
This is true. How does it fit in? I mean, it doesn't. I don't know. My Twitch stream is very separate from my work. I guess except for like the communication aspect. Like there is a lot of I do a lot of video for work as well. And I actually I haven't been there super long, but I remember doing my first video, and the one of the producers came back to me and was like, "Hey, like, have you been on video before?" And I was like, "Yes, I have actually." So a little bit of crossover there, I guess, insofar as like how I carry myself on camera, but otherwise quite separate.
That's still really cool. Could you tell us a little bit about the stream as well, just so the folks at home know? I mean, obviously in the show notes it'll be all your Twitch stream and your YouTube page, but like, what is it that you do there, and what got you into the streaming as well?
Yeah, so I started streaming when I was waiting for immigration paperwork to go through. So I am Canadian, but I live in the US, and there is a time when I was under a visa status where I was unable to have income. And so while I was waiting for that to come through to start work, I was like, hey, I there's a whole bunch of video games that I want to play. And I had just kind of discovered what Twitch was. And I was like, maybe people would want to watch me do that. Because also I grew up in a household that didn't have video games. So through friends, I'd learned about a bunch of different retro games that I definitely wanted to check out that sounded up my alley. So I started there. I started with three games that I wanted to play. I wanted to play Chrono Trigger, I wanted to play Earthbound, and I wanted to play Final Fantasy VII because I'd never touched them before. So that's where I started, and that's kind of still what I do. Almost everything on my stream is a first playthrough of a retro RPG. I am a turn based combat lover, um, but I take suggestions from my community. They vote on what I play, and they have effectively given me an education on on retro gaming and retro RPGs, and gotten to relive their nostalgia through me and through my fresh eyes on their favorite games.
What a good three to start with, by the way. Just as like
Yeah.
that is a really good three to start with. Did you
It really
have
was. It was solid.
did you have a favorite of those three where if you're looking back on it, you're like, you know what, that was the one?
This feels like a dangerous question to answer. I think for me, of those three, it's Chrono Trigger.
I don't think there's a wrong answer on either of them. That's really really cool. So and then you also became partner there now on Twitch as well. You're a member of the Twitch Women's Guild too. So
Yeah.
you're making moves in every avenue, which is really really exciting to see. Um, yeah, I just like you're extremely busy and just doing a lot of incredible things. I just again want to thank you for taking the your time out of your busy day to talk with us about this journey because this is just it's so cool to learn about and having shared your experiences. Do we still have time for a few more questions from the audience as well?
Yeah, I'd be happy to do that.
Folks were wondering, as you're working with these animals and you're integrating changes in their diet, whether, on any one of these animals, is there, what overlap do you have with the USDA in terms of then having to label if these animals are in any way genetically modified through their feed, like because you're modifying the feed that causes genetic changes in their gut? Or was that something where that's later on in the product development line of things that you never had to really think about?
Yeah, so because these were all research animals that were not destined for the human food consumption, that was never a thing that we had to worry about. Um, I think that would happen a lot later once it did become an established product, and if those animals were going to, like, for human consumption. Um, there was a consideration when we were giving the bacteria to the piglets, so that bacteria is, it has GRAS status, or it is generally regarded as safe. So that is something that we thought about if that had been the direction that we went with that. Um, but no, none of these research animals did we have to worry about withdrawal times or anything like that.
Cool. So that would have been a much more like it sounds like it's already been kind of accounted for with like you saying grass safe if it did get integrated in some way. It's really neat then. It's like maybe it was a downstream issue then in terms of like licenses or permissions or labels on foods. Like it's almost like you're not going to look at that until you've proven that this methodology works and it's worth the time to invest from that particular aspect, right?
Yeah, exactly. Any of the research that I did would have been like steps before that.
So it's so cool to see like and hear about these steps too. Again, like I'm nerding out and thinking about these are all things that are going on behind the scenes every day and that we just don't actually get to hear about them, but they are critical for, food security and food development that, you were on the front lines of. And it's just so cool, like, pigs, chickens, and cows that you had the experiences with. Since it is our Women in STEAM month, we've had questions too of if you being a woman in STEAM, did you find any difficulty in this field and any challenges that you had to overcome? And if so, what advice would you give to the next generation of women going into this field?
I would say that generally I have felt very lucky, um, insofar as not having experienced a lot of discrimination. I also think like maybe I approached it a bit naively as well. And by that, I mean, like, I was never told at home by my parents that there was anything that I couldn't do or that I was other because of my gender, or that played an impact at all. So it was never something that I was actively thinking of as a youth, going into university. One thing that I will say is kind of evident, and it's just about like working with cattle, and I think it is because cattle are large. And traditionally, when you think about, if you think about like a rancher, they're probably male. So to have a woman working with cattle was maybe the only point where I noticed something a little bit different. Now, that being said, like I am tall. I am 5'10". I am not a small person. So I think like because I have that physicality to me, it maybe it affected me a little bit less. Um, but like my PI and my PhD was female. The my supervisor when I worked with cattle was also female. Like I was surrounded by these strong female presences who never led me to believe that there was anything I couldn't do. And I think surrounding yourself with people like that is very important.
That's really cool to hear that there wasn't any kind of like at least strong negativity about it, because you always hear these nightmare stories. It almost seems almost field specific, where it's like in this particular field you're going to have a lot of difficulty. We've actually learned this series that in archaeology it seems like it's mostly women and
Mm,
supporting one another, and it's like primarily women in the field, and it's really cool to hear. It's like, yeah, it's everyone's on the same page supporting one another, and we're not having this kind of almost a need for the discussion almost, because everyone's just doing well, which is on it like the end goal, right? That they're you don't have to have these conversations because everyone is on the same page.
mm, exactly. Yeah.
But it's again, as we're wrapping up, Leandra, it's really thank you so much for your time and sharing your experiences with us and the stories of your research. I had so much fun nerding out and asking questions, and the chat too is saying like how cool your research was and just how much fun it is to hear about. Um, One thing that we've been asking all of our guests as well is that, you know, you yourself are a leader in your field, right? I would argue that you have looked at more animals than most of us ever will, right? Both on the outside and the inside, developing product, looking at food security and trying to develop product to help people, right? Acquiring food, going, transitioning from the vet pursuit to being a research scientist, like with holding a PhD, working in animal nutrition and the implications of the nutrition. Many would say, and I would highlight that you are an inspiration to many women in STEM, right? As someone to look up to. And so, what does it mean to you to be not just a woman in STEM, but a potential inspiration for the next generation?
I mean, it means a lot. And I'm very grateful for you to be hosting a space like this where we can tell our stories and potentially inspire other people to just, I guess, to think about options or what they're able to do, or what they're capable of. So, yeah, I mean, it maybe feels like a lot of responsibility, but also it's just really cool.
I think it's really, really cool. And I can't, it's so easy to want to further cheer you on and to be successful because you can see the passion in your eyes, the love of what you do, this sharing this information with us. And it was so easy to talk with you tonight. It was an absolute joy and a privilege, Leandra. So thank you again for joining us. What would you like to promote? Please share everything, anything upcoming, links that people should click. We've got your YouTube and your Twitch page in the show notes as well as in the chat. Please, anything and everything. The floor is yours.
Yeah, I mean, if you want to come hang out in my Twitch stream, I don't have a solid schedule right now because I kind of just stream when I have the energy for it. Maybe some of you can relate to that. But you're always welcome. We do chat science sometimes. I used to have a science routine. Maybe it'll come back where we'll talk about kind of what's new and what's interesting. Um, and yeah, my YouTube just hosts all my VODs. So if there's a retro RPG that you really like, maybe I've played it, and maybe my playthrough is on there. But yeah, come by the stream. I'd love to meet you. I'd love to know what you love.
Awesome. Thank you again, Leandra. Time, expertise, just all of it tonight. And we've already had from chat that folks are, yeah, I already followed. Looking forward to it. Um, so thank you again. Have a wonderful rest of your evening and keep on just being you.
Awesome. Thank you so much.
Thank you, Leandra. Have a great evening.
Science is for everyone,
Bye.
all backgrounds, all ages, all genders, and all walks of life deserve an education. Come be one of us.